Hello and welcome. I want you to just take a look around you for a second. On every single surface you can see, on your keyboard, your phone, even inside your own body, there's a war raging. It's a battle being fought on a completely invisible battlefield, a hidden world we're really just starting to get a handle on. So today, let's dive into the absolutely fascinating field of microbiology. Think about that for a second. What if the vast majority of life on this planet, something like 60% of all organic matter, was stuff you couldn't even see? Well, it's not a what if. That's the reality. We are massively, massively outnumbered by life forms that are completely invisible to the naked eye. It's a staggering thought. But here's the thing about this invisible world. It's not just good guys and bad guys. It's way more complicated. These microbes have this incredible duality. On one hand, they're our allies. They're essential for making foods we love, like cheese and yogurt. They produce life-saving antibiotics, and they're crucial for recycling nutrients in the environment. But they are also our adversaries. They're the pathogens responsible for everything from a simple cold all the way up to devastating global pandemics.
So how did we even find out about these invisible entities in the first place? Well, it took centuries of hard work by some of science's very first detectives who slowly, piece by piece, started to solve the mystery of a world that was hiding in plain sight the entire time.
The investigation started pretty slowly. Back in the 1600s, you had Robert Hooke noticing some fungus. A little later, Anton van Leeuwenhoek, this guy who built his own amazing microscopes, saw what he called animalcules, these tiny little creatures zipping around in a drop of water. But the real game changer, that didn't happen for another 200 years. That's when Louis Pasteur comes along with his totally radical germ theory, the idea that these tiny organisms are what actually cause disease. Then Robert Koch came in and proved it, connecting specific microbes to specific illnesses, and with that, he changed medicine forever.
Okay, so now that we know they exist, let's meet the main players in this whole invisible drama. I want you to think of this as a kind of police lineup, a real rogues gallery of our primary microbial suspects, and each one has its own unique way of causing trouble.
Now, to properly identify our suspects, we first need to get one fundamental thing straight. In the world of cells, there are basically two main types. On one side, you have the simple guys, the prokaryotes. Think of them like a tiny studio apartment. No nucleus, everything's just kind of floating around in one open space. Bacteria are like this. Then you have the eukaryotes. These are the complex cells, like the ones that make up fungi or plants or even you and me. These are like mansions, with a central nucleus as the main office and all these other specialized rooms or organelles doing different jobs.
So, let's meet our first and most common suspect, bacteria. These are your classic single-celled prokaryotes. Their game plan is incredibly simple. Find a good environment with some food and just multiply by splitting themselves in two. They are the ultimate survivors, and you can find them pretty much everywhere on Earth.
All right, suspect number 2 is a completely different kind of beast. In fact, viruses aren't even technically alive. You should think of them more like cellular pirates. They're what scientists call obligate intracellular parasites, which is really just a fancy way of saying they are totally helpless on their own. They absolutely have to hijack a living cell to survive and make more of themselves.
And here's exactly how that hijacking goes down. First, the virus latches onto one of your cells. Then it injects its own genetic code inside. From that moment on, it basically turns that poor cell into a zombie virus factory. The cell's own machinery is forced to create and assemble thousands of new virus parts until it gets so full that it just bursts open, releasing a whole new army of viruses to go and infect more cells. It's brutal.
Next up in our lineup, fungi. Now, unlike bacteria, these guys are eukaryotes, just like our own cells. They can be anything from single-celled yeasts to complex, fuzzy molds. A lot of them are totally harmless, but some are what we call opportunistic. They basically just hang around, waiting for your body's defenses to be down, and then they strike, causing fungal infections, which have the clinical name mycoses. And our final suspect is the most mysterious and, honestly, the most terrifying, prions. These aren't even cells. They're not alive. They're just infectious, mangled proteins with no genetic material at all. What they do is cause these fatal brain diseases, like Creutzfeldt-Jakob disease, by finding normal proteins in your brain and forcing them to misfold into the same dangerous shape. It sets off this unstoppable, deadly chain reaction.
Okay, so with such a diverse and dangerous rogues gallery, how do scientists and doctors actually figure out which specific culprit is causing an infection? This is where the microbiology lab turns into a forensic unit, using some really clever techniques to unmask the perpetrator.
One of the most fundamental forensic tools is called the gram stain. It's a surprisingly simple but really elegant procedure. First, you apply a purple stain, called crystal violet, to the bacteria. Then, you wash it with alcohol. And finally, you add a red counterstain. The color that the bacteria ends up holding onto gives us our first major clue about its identity.
So, what does the color actually tell us? Well, if the bacteria stays purple, we call it gram-positive. That means it has a thick, simpler cell wall that holds on to that first dye. But if it turns red or pink, it's gram-negative, which means it has a thinner, more complex wall. And this one single distinction is absolutely vital because it helps doctors choose the right kind of antibiotic to knock out the infection.
But just staining them isn't the whole story. The next crucial step is to actually grow a pure sample of the suspect in the lab. That's the culture. Then, scientists take that colony and expose it to a bunch of different antibiotics to see which ones actually kill it. That's the sensitivity test. And it's how you make sure you're using exactly the right weapon for the job. All right, we've identified our enemy, we know its weaknesses. So now, how do we fight back? This is where we get to the climax of our story, developing and deploying a real battle plan to defend ourselves against all these invisible threats. And make no mistake, the stakes could not be higher. This number you're seeing, 99,000, that's the average number of people in the US who die every single year, just from infections they picked up while getting medical care. This invisible battle has very real and very, very serious consequences. Fortunately, we aren't defenseless. We're all walking around with this incredibly sophisticated, multi-layered defense system built right in. Your first line is all your physical barriers, things like your skin and mucus. If an intruder gets past that, your second line, the innate immune system, kicks in with general-purpose weapons like inflammation and white blood cells. And finally, you have your third and smartest line of defense, acquired immunity. This is the part that learns to recognize specific invaders and creates custom-built antibodies to hunt them down and destroy them.
And this is where we get really proactive. We can actually train that third line of defense. That's exactly what a vaccine does. It's like giving your immune system a cheat sheet. We introduce a safe version of the enemy. Maybe it's weakened, maybe it's killed, or maybe it's just a tiny piece of it, so that our immune system can learn how to defeat the real thing without us ever having to get sick in the first place. But there's a plot twist. A big one. The enemy is adapting. Bacteria are evolving resistance to our best antibiotics, creating these terrifying superbugs like multi-drug-resistant tuberculosis. The weapons we relied on for decades are becoming less and less effective, and it's forcing us to get creative and find brand new strategies. This is exactly why our most fundamental battle plan is so unbelievably critical. In every hospital, every clinic, these standard precautions are the foundation of defense. We're talking about simple things: washing your hands, wearing gloves and masks, handling needles safely. It's not always high-tech, but these simple actions are incredibly powerful tools to stop an infection from spreading before it even starts. This quote really gets to the heart of why those precautions are so non-negotiable. It points out that the healthcare workers themselves, the very people trying to help, can be an exogenous source or an outside source of infection. Any tiny break in sterile technique exposes a patient to a risk that was, in most cases, completely preventable. It all comes down to constant vigilance. So, what this all really comes down to is this. The fight against microbes isn't a war we just win one day and it's over. It's much more like an ongoing, dynamic relationship that we have to manage. It demands constant vigilance, it demands research into new strategies like phage therapy, where we use viruses to kill bacteria, and it demands a relentless effort to better understand our invisible adversaries. Which leaves us with one final big question. Microbes have been evolving on this planet for billions of years, and trust me, they are not stopping now. In fact, they're mutating faster than ever. So the real question is, how will we adapt? What new forensic tools, what new battle plans will the next generation of scientists and doctors have to invent to stay one step ahead in this never-ending invisible war?